Hello, everyone, and welcome to the Midweek program at the Raulston Arboretum. My name is Blake, and today we are on the, I guess we're on the mixed border. We're in the middle of a drought right now. You may be able to hear some sprinklers in the background. This border got irrigated yesterday, so we are sitting here just so we don't get sprayed, really. We got a program for you all about plant hormones today. We've got our research technician, Villani, here. He's going to be giving you an overview on plant hormones. I gotta say, I'm excited about this. I was fully expecting Evan to show up with a PowerPoint presentation on a flash drive. Instead, he showed up with a flat full of plants. So it should be really fun. It should be really interesting. And hey, we'll get to look at some plants while we do that. Those of you watching this on YouTube, please like this video and subscribe to our channel, and feel free to leave a comment down below. And with the announcements out of the way, I will pass things over to Evan to give us a nice little overview on plant hormones.
Hello, dear viewers. I'm brought to you today with plants. This is probably the most time I've spent prepping for Midweek, and it's probably the least practical Midweek you may ever watch. I want to set the stage here by saying the point of this program isn't for you to come away with big takeaways on things you should do to your plants. It's more of the world inside of a plant is so astounding that maybe you just sit here and you listen for a while and you think wow, it's amazing that this even works. Because I think we here at the Arboretum think of plants as stories. Sure. And this isn't just a single plant that you're looking at here, but instead it's the story from the beginning of creation until now of this plant going through, not this plant, but all of its ancestors all the way back to a single cell, and what environments they went through and how they adapted to those environments and how they found a way to survive. And it has somehow brought you this here today. And this only exists right now, and in a million years it'll change, and it won't be the same. Absolutely. So I think that's amazing. And we are going to get into it with a couple fun facts. So fun fact number one, plant hormones have a remarkable similarity to animal neurotransmitters. So it seems like about 1 billion years ago, all of life agreed that the fastest way to spread information internally is through small diffusible particles. Okay. Which cells can pick up on or see and then react to. Okay. So, number two, plant hormones are remarkably ubiquitous. So they are present in all of the things that we consider plants, which are angiosperms mostly. They go back to ferns, and a lot of them go back to bryophytes and mosses, and all the way up until they just came out of being algae. So 500 million years ago, a lot of these hormones developed, and we're able to kind of see them in action today. And when I talk about angiosperms, those are flowering plants. And a big question I kind of have been running into recently is, why are there so many angiosperms? Why are they so successful? They're everywhere. Sure. And it's sort of because they're adaptable. They're able to react to the environment, and they have a vascular system, so veins, so they can translate. They can move resources over a distance within themselves. They can flower, which is a kind of new way of reproducing themselves and spreading versus with animals and on seeds. And then they have these hormones, which we'll talk about today, which set them up for doing a lot of different things in response to the environment. Okay. And this is a pretty young field of study, actually. It's really less than 100 years old. So this is fun and exciting. Okay, I would like to go over some vocab so you at least get some sort of definite knowledge out of this. That seems like a good plan. And you did throw the word neurotransmitter out there momentarily. So yeah, let's learn some words, shall we? Neurotransmitter, you can kind of see the word neuron in that. And neurons are all the ways that our bodies as animals send signals. Okay. Through electric impulses and hormones. Well, it's the way that an animal organism can, within its own mind, send information from one part of it to another part of it. Yeah, how do I know I'm touching this table? Right. Right? For sure. There's a signal that travels through here. There's a signal from the hand up to the brain, and then from one part of the brain to the other part of the brain that actually experiences that. And so
for sure. We're communicating with chemicals within our cells.
Right, because the hand alone does not know it's touching the table. The hand doesn't know it's touching the table until the brain tells it that the hand is touching the table.
No kidding. Gosh.
This will come into play later. We'll bring the first vocab word in, tropism.
Okay.
Tropism is a plant response. That's all it is.
All it is.
Phototropism.
Yes.
A plant's response to light.
Okay.
Gravitropism, a plant's response to gravity.
I'm seeing where this is going.
Chemotropism, a plant's response to chemicals, mostly nutrients. That's how roots know where to grow, except if it's not water.
Okay.
And there's with vines, there is, I forget what it's called, but it's the tropism of touch. Vines know what they're touching. They know what to grab hold of.
Sure.
And it's the same thing. So the plant doesn't know until something tells it to the whole system what to do.
Sure. And then it elicits a chemical response so that the plant can then indeed know what's going on.
And react to it.
Yeah. Awesome.
I wonder, I didn't bring a vine.
That's okay. There's a Kibia quinata growing on that trellis over there.
Yeah. And it'll sort of know to grow in the direction where it feels something. It'll feel something and then elicit more growth there so it can continue to grab, and then so on and so forth. And it also, at the same time as it's feeling touch, it's feeling light. So it makes sure that it's getting both. And then it grows towards the light along a structure. Another way plants know up from down is through polarity.
Okay.
So polarity is based sort of from the trunk. So it knows that everything this way is out, and it knows that everything this way is also out. So there's, for the plant, there's only outwards from the trunk or inwards from the trunk.
Okay.
And so when it's moving resources through itself, it says, do I want to go towards the trunk or away from the trunk? Essentially. There's also only 2 places where things are really happening in a plant in terms of hormonal activity. That's a little bit of a simplification, but it's the tips, the growing tips. So tips of roots that are actively growing and tips of shoots that are actively growing.
Okay.
And then there's also, because we know that as trees grow, they get wider, right?
Yes.
So there's a ring of cambium, which is also undergoing a lot of change. And when I say change, and again, I'm sorry, this might get overwhelming, but when I say change, it's the cells themselves deciding what to be.
Okay.
So in these places, there are a lot of undifferentiated cells.
Interesting.
Which just means raw material cells that are ready to be anything. They can be roots, they can be leaves, they can be flowers.
Yes. Because keep in mind, every individual cell of a plant contains all of the information, all the genetic information. So any individual cell could be a root, it could be a shoot, it could be a leaf. It contains all of that information, so it has to figure out what it is.
Mm-hmm. And that's one of the ways that we do propagation, and how plants reproduce themselves sometimes is vegetatively. So if a part falls off and it lands in a nice location, all those undifferentiated cells can then decide to become new organs.
Yes.
So roots, shoots, leaves, and flowers to make a whole new plant from exactly identical to the plant what it fell off of. And we do that through propagation. We manage that. Plants also do photosynthesis. Photosynthesis is the intake of light and carbon dioxide to create sugars. And in order to do this, plants on the underside of their leaves, I don't know if you could see, you probably can't see, but have stomata, which are itty bitty openings, which gas can go through. It's because they want to take in carbon dioxide. But at the same time, they have open pores. They release water. That just is a way. That's the way that it is. Plants are wet inside because they're alive, and so by opening up their pores, they will release water. So that's one of the things a plant has to balance. How much carbon can I take in? How much light can I take in? how many sugars can I produce without losing too much water, and
For sure.
and how much sugar do I have in me right now to
Yeah.
kind of kickstart other growth processes.
Because they need to open those stomata to do the photosynthesis, to make that food. But when you do that, you're losing water, which can be a problem. So you're saying balance, like the plant has to regulate these things. So how does the plant regulate these things?
So the plant regulates these things through hormones. Are we ready to jump into it?
I think so.
Okay.
So far, we've gotten a bit of an overview of the physiology, and it's just like, well, what does this have to do with hormones? I'm assuming we're getting there now-ish.
Let's jump right into it.
Okay.
So the hormone in charge of opening and closing these stomata is abscisic acid.
Okay.
Abscisic acid is unlike the other ones. It's not like a hormone group. There's just one molecule that is abscisic acid. There's no differences. It's a very universal molecule. The molecule looks the same across all plants.
Nice.
When a plant is experiencing water stress, there are genes that produce abscisic acid.
Okay.
And when enough abscisic acid is produced, it runs to the stomata. So from where it's produced, it'll make its way towards these stomata.
Yeah, you were talking about the whole story of all of evolution and how these angiosperms, they developed a vascular system. So specifically to help with the transport of these hormones, as well as sugars and water and that kind of stuff. But it's all about that communication network within the plant, getting the right chemical signals to the right places at the right time.
Yes. And so abscisic acid will run to the stomata and then activate a shift in ions.
Yeah.
Which all that is to say, it makes the stomata swell up and close again.
Yeah.
So this does mean for the stomata to swell up, the plant does have to have a certain amount of water left. So that means the plant can never expend all of its water, or it loses that leaf. And one thing you're seeing here, which is also the result of abscisic acid, is leaf drop.
Yes.
Another word for that is leaf abscission.
No kidding.
So the plant decides sort of when to cut off elements of itself when it's no longer worthwhile. So there's some calculations the plant does. There's enough hormones bouncing around that tell it, this leaf is no longer worthwhile. I'm spending too much water.
For sure.
And I'm not getting in enough resources. So leaf seals, the abscisic acid seals a little wound underneath the leaf, and it drops off.
And we're seeing that in the garden right now. We're in the middle of this big drought. We actually had a rain event like a couple weeks ago, and Evan was just talking about how they need that little bit of water to even close their stomata. They also need that little bit of water to form the abscission layer. So
that's right.
We had a whole lot of drought, then we had a little bit of rain, and over the next couple of days, we saw a bunch of leaves falling from the trees because they were able to form that abscission layer and actually lose those leaves that were there. It was too much for the plant to keep those alive. Like it was wasting too much water while it was trying to photosynthesize. So the plant decided, hey, we'll get rid of these extra leaves in this stressful time, and we're going to do it with the power of abscisic acid.
That's right. And if a plant doesn't do it slowly, and some like conifers, they just die all at once. So all the leaves go brown, they hang on, that's it, it's done. There's no recovering. It ran out of juice.
Yeah, for sure.
Yeah.
The permanent wilting point is a phenomenon in biology and plants. It's the point at which the thing is wilted, and it is that wilting is permanent, and it's defined as once the plant cell is no longer has water in it. Like the plant needs water in the cell to make sure that everything within that cell still functions properly, and without that water, things just don't work. Life ceases. That's the permanent wilting point.
And abscisic acid works primarily with two other hormones. One is auxin.
Yes.
And we can jump into that. And the other is gibberellic acid.
Okay.
It works with auxin.
I feel like gardeners may have heard of auxin.
You may have heard of auxin because it is a very common hormone in plant propagation. The interesting thing about that is that plants produce auxin all the time. super rapidly and it moves around. It moves around kind of slowly, but it's everywhere. Every growing tip is producing auxin, and it's mostly in the shoots, so where the leaves are. But auxin has a really short half-life, so it breaks down really quickly. So this makes plants really sensitive to changes in the level of auxin. Auxin is kind of like the overarching hormone.
Okay.
It interacts with thousands of genes.
Yeah.
And basically every other hormone.
Yeah.
It's kind of like it's just everywhere, and everybody has to know auxin.
For sure. And everybody knows auxin as the rooting hormone, but the interesting thing there is we just said that it's made in the tips, it's made in the meristems, but so it's made in the meristems and then sent all throughout the plant, all the way down to the roots, which then, so it's really like the concentration gradient of it within the plant that really determines how it functions, more or less.
It's where's there a lot and where's there a little, and the plant will respond based on that.
Definitely.
So I'll talk about how it interacts with abscisic acid. So abscisic acid closes off things, right? So when it's also in drought stress like this, what's happening underground, which we can't see, is abscisic acid is entering the roots as well. And so it'll close off a lot of root pores, but auxin is still going through these roots. And what ends up happening is in all these pores are closed except for the very tip of the root. So all I'm trying to say is that all of the energy in roots becomes, instead of them spreading out where they are and dominating a region which already the plant knows doesn't have enough water, everything is shot into the root tip. So it's going to go as far as possible in order to find water and nutrients, but just mostly water. And this does have a practical application. In drought, you don't prune the tips of a plant. You don't prune a plant, really, because, well, one, in order to close off a wound, it needs water and to seal up healthily. But two, those tips are producing auxin, and it's auxin that's making roots root more. So if you prune a plant in drought stress, you're reducing its rooting capability, which is also why we don't prune fresh plants. So when you plant a plant, we like to leave it be for at least a year, unless there's like dead or decaying material on it. Because leaving those actively growing shoot tips actually make the root tips grow more. But then when a plant is happy and established, you can do whatever you want. Talking more about auxin, it has a really weird thing. We have this podocarpus here. It's growing straight up. It's growing straight up because in the nursery, it has enough light from all directions. However, we'll see, especially in woodland edges, and if you've ever watched sort of a time-lapse video of a seedling growing, that a plant will bend towards the light. And auxin is the reason that happens. So auxin is produced in the tip and slowly sort of percolates down, and it avoids sun. So if the sun is right here, all the auxin will move to this side of the plant. And then if that happens, so over the course of the day, if this plant is in full sun, the sun will move around the plant and there's not really going to be a big effect. But auxin is in charge of cell elongation, so expanding cells. So if all the auxin is on this side and the sun is only ever appears on this side, so like this is like a wooded area and it's all in shade and the sun is here, then all the auxin will sit on this side and slowly continue to expand this side really fast, and the stem will bend towards the sun.
For sure.
And that's the primary thing behind phototropism, so how plants respond to sun.
And that's something that you can actually watch in real time if you have the right plants. I've got a Hoya carnosa I've got a Hoya carnosa and a Hoya pupacalyx growing on my desk. And the Hoya pupacalyx, it sends up a little vine, and throughout the day, it's slow. You can't watch it in real time, but it's like you work for a little bit, the vine's this way, you go do something, you come back, it's like this, and you realize, wait a minute, you've been moving, and you can watch it throughout the day. Really, it's slow, but it's happening. You can see it happen over the course of a day. And
vines and thin-stemmed plants are more sensitive. They have there's less cells to move around, so every bit of change is more change. We'll do a couple more interesting things about auxin and then move on.
Beautiful.
So auxin, interestingly, affects its own production and breakdown and reception. So when I say reception, that's communication, how a cell perceives the auxin. And that is usually on the outside of a cell, there are receptors. You may have heard of this in sort of like, we learned about viruses, but there's a receptor, and something will bind to that receptor. There's a receptor specific for auxin on the cell.
For sure.
And then the auxin binds to it, and then something happens in the cell.
Yes.
However, there's 2 receptors of auxin that are in like weird locations.
Okay.
So one of them is in a hydrophobic gap.
Ooh.
So that means like, it's like nothing that likes water can pass through that gap.
Okay.
So there has to be enough auxin to fill up that gap, and then an auxin molecule will make it across the gap and bind to the receptor. And then I'll say the other one, which is pH derived. So auxin is a little bit acidic, and some cells are in a more basic environment. But again, if there's a flood of auxin, it'll change the pH to a more acidic environment, and that allows it to bind to the receptor. So in these 2 ways, there has to be enough auxin to elicit a response.
That's awesome.
Yeah. So it can't just be like a little bit of this auxin, which is like everywhere at all times. It has to be, there has to be a big change in environment or a stress or something that tells it, okay, this is worth reacting to. And then as auxin is very important, it's also involved in cell fate. So auxin is the thing that from the beginning to the end of a cell's life tells it what it's going to be and how it's going to die.
Okay. That's pretty heavy.
Right? So all these undifferentiated cells that are everywhere, their development is based primarily on an auxin gradient. So where the auxin is within the cell, I don't even fully understand, but that's how it goes.
No, this stuff's really complicated. Like plant nutrition was legitimately the hardest class that I had in college. Like it's serious business because it's not just the question of what does this hormone do, but there's also interactions with that hormone with every other hormone. And it just gets complicated.
It's a big living system.
Yeah, absolutely.
And the other thing it's half in charge of is cell division. And that allows us to move into cytokinin. So auxin and cytokinin are both together in charge of cell division. However, auxin favors root production and cytokinin favors shoot production. And this was discovered, if anyone has heard of tissue culture, which is the art of sterilely reproducing plants, researchers had to find a very fine balance between cytokinin and auxins, that there was a little bit of root production to sustain the plant, but they mostly want shoot production to make the plant productive because they're growing, they're making more of it. So they want there to be more of it and not just a bundle of roots, essentially. And Scooge, which is a lot of tissue culture is based on this mixture called Muskogee and Scooge. I'm probably saying that horribly, but they're fine. 2 of the primary researchers who figured this all out. And Scooge somehow got this from herring sperm, so fish sperm. Had this hormone called kinetin, and kinetin produced a lot of shoot growth in plants. So that's, again, similarity between animals and plants is
no kidding.
Just why is that?
It is wild.
Yeah. And then from there, they were able to say, like, they eventually did more research and found cytokinins, which is a group of about 7 hormones that produce shoot production in plants. It is present in agrobacteria, which is one of the main ways that we do genetic modification, essentially, is bacteria mediated. And so when you see these huge galls on a tree, if you've ever walked past that, that's probably agrobacter. And that's a bacterial infection. And what it does is the bacteria has genes that it sends into the plant.
For sure.
And we use that technique to get genes into plants. That's the thing. But the bacteria sends genes into the plants that tell it to overproduce cytokinin and auxin. And so when there's too much of these hormones, the plant just expands right then and there. There's no time to translocate or move these hormones. They all just get bundled up right there and they have to get used up. There's just so many of them that the plant has to respond. And it forms a huge gall. and
Yeah.
the bacteria just loves that gall. It just sits in the gall. It sends in these genes to the plant that says, produce me some sugar, plant. And then it just sits there and eats the sugars made by the plant and reproduces itself. And that's the life of Agrobacter. So.
Yeah. But it's something that scientists can harness and use with the bacteria's ability to inject genetic material into the plant that it's living on. So like we take that's how we insert genes when we're genetically modifying organisms, doing, I guess, bioengineering. Is that maybe the word you were looking for?
Something like that.
Something like that.
Yeah.
There's a word for it that is not in my head now either.
It was probably that. I think so. There's so many fun facts. I'm so sorry.
Yeah.
So the other way it interacts with nitrogen or auxin is apical dominance.
There you go. I was going to ask. We covered that.
Auxin is the thing in charge of apical dominance. So that's very, very clearly present in this. It has one main shoot going up. That helps. And apical dominance helps tell the plant which way is up, and then every other growth is sort of sublimated to it. That's not the right word either.
Man.
It's lesser.
Yeah.
It's,
subordinate.
Subordinated.
Yeah.
Yes. All the other growth is subordinated. This is the top. This is the hierarchy of the plant. I'm the dominant apical meristem.
Yeah. 'Cause here's the thing about plants like this. This is a leaf. The bud of these is just below that leaf, and below every single one of these leaves, there's a bud just below there. And none of those buds are breaking because this bud at the top has auxin and it's exhibiting that apical dominance. If you were to cut this off, if you were to cut this to there, then whatever leaf is left, this is the first one left, that bud just below that leaf, that's what would grow out on into infinity. But the reason all these buds on all these leaves aren't just shooting off in every direction right now is because the auxin that's being produced in the top of this plant sending it down.
Right. And so if you remove that auxin, the cytokinin takes over and all the other shoots produce.
Yes.
So that's
hormonally,
that's what's happening.
And we go back to the balance between the two hormones. It's not necessarily that you have an abundance of one and an absence of the other, but it's the relative proportions of the two.
And I don't know, can you see this, Tessa, where my hand is? If you can zoom in where my hand is, you see like this lighter colored stem?
Yeah.
That's more recent growth. And right here, that little black bit, that was originally the dominant stem.
Yeah.
But either it died or got pruned back.
Yeah.
And then the next stem below it became the dominant stem. And look, it's already as thick as the branch below it.
Right.
So that's just how the plant is responding to that.
For sure. And it's cool the way a plant like tells its story like that through the way, just by it being what it is, and how you can read what is the new growth, what's the old growth, how this used to be apart, but now it's moved. You can see across time and space just looking at an individual plant. It's really an amazing thing.
It is. It is. And then last thing, cytokinin is also kind of in charge of nitrogen. It tells the plant how much nitrogen you have. And it also is responsible for making those little galls in the roots of legumes and things for symbiotic bacteria to live in. Okay. So that's just awesome. Okay. And now we're on to gibberellins. And gibberellins are the other hormone that interacts with abscisic acid. It's the opposite. So gibberellins are a growth promoter.
Okay.
And they're also what kind of sometimes tell seeds to germinate.
Sure.
Abscisic acid is what slows down metabolism. It tells everything to stop. It says, we're stressed. We need you to be able to survive this period of times. And gibberellins is the green light, the go, go, go. G for go. And it is also the most diverse group of things. There's 135 members. And it's shockingly easy for the plant to switch these members around, and they all do different things. I have this sheet of paper here. This is confusing. It's just a little bit of organic chemistry, but it's showing you that once a gibberellin molecule is produced, it can switch between 20 members pretty easily.
Sure.
That's all I wanted to show you. And it's kind of remarkable in chemistry because the enzymes which it uses to do this, and enzymes are just proteins that make things happen.
Yes.
The enzymes are called promiscuous because they can accept multiple substrates and they're multifunctional. Okay. So like one enzyme can take in 3 different forms of gibberellin and form them into like 8 different forms of gibberellin. That's
Okay.
The numbers on that. Don't quote me on the numbers. But that's just why.
Just an example to show how complicated that things can get.
Exactly, and it's weird. It's weird that happens. I mean, most enzymes do one specific thing, and that's it.
Sure.
It doesn't, like, it's hard for a plant to control it. It seems like that would be something that easily gets out of control. How do you manage something that does 8 different things? I don't know. I'm not a plant. They're in charge of shoot elongation, so the plant getting bigger, growing out, and starch breakdown.
Okay.
So we see on this plant, we have a lot of little new growth up and along the stem. This plant is happy. And then if there is temperature stress,
Okay.
gibberellins are what also tell the plant to break down your reserves of sugar and starch into sugar so that we can do more things. We're stressed. We need to start it's like you're gonna run, so your body in response to running breathes more heavily.
Yeah.
Kind of like that. And
For sure.
well, the hormone is what's prepping your body to breathe more heavily.
Yeah.
Specifically. Yeah.
Yeah, and like starch is a way for plants to store energy.
Yes.
And so, but it needs to be transferred from starch to more simple carbohydrates so that it can actually be used as energy. So the gibberellins help that process of converting the stored energy into a more useful version of energy.
Yeah.
So that it can go, because G for go.
G for go.
G for go.
And the last fun fact about gibberellins, and then I'll move on, is that one of the main receptors, GID, is sort of shaped like a mousetrap.
Okay.
So what will happen is once a gibberellin molecule enters it, the lid of the receptor molecule changes and traps it in there.
That's crazy.
So it's like, okay, we're good, we're going. We got the response, we got the green light. I'm keeping the green light until I'm ready to let go of the green light. It's just wild.
Yeah.
And when you see dwarf plants, a lot of those are not able to produce gibberellin.
Interesting.
Or they have limited gibberellin production.
Okay. Could you elaborate on that a little bit? So like if they have limited, so it's just they're dwarfs, so they don't have enough gibberellins, so that means they can't extend long enough.
Gibberellin is in charge of shoot elongation.
Yeah, okay.
So if the shoot doesn't elongate, you just end up with a more densely stacked plant.
Gotcha.
And we call that shorter internode space.
Yep.
So instead of there being these long, like this, has this internode space. So there's a node where leaves come out, and there's all that stem, and then there's another node. But this is very different. So the internode space here is like, it's in millimeters. You know, there's a leaf coming out, and then there's a leaf coming out, and then there's a leaf coming out, and then there's a leaf coming out. So there's a lot less stem elongation on this, so you end up with denser foliage as a result. Next. Ethylene.
Okay.
You guys may have heard of this. This is a very simple molecule. It is just 2 carbons next to each other.
Yeah.
And ethylene is a gas. So within a plant, it travels as this thing called ACC, which is just a shorter name for a molecule that carries this, and it's like ready to break off. It's like, I could break off the ethylene at any moment, but it holds on to it. And in this form, it's a solid. It can be moved through the plant.
Sure.
So that it's released in the right location.
Gotcha. So it's not traveling through the plant as a gas, it's being translocated as a solid, but then when it gets where it needs to go, then it can off-gas the ethylene in how it needs to do it. So what is it used, that ethylene, for?
Ethylene is sort of the maturing hormone. So most commonly, we see this with one bad apple spoils the bunch.
Yes.
So fruits are they produce a lot of ethylene, and they're very responsive to ethylene, some of them.
Yeah.
And so as they ripen and mature, they release ethylene, which causes them to ripen and mature.
Yes.
And it's sort of the whole plant timing itself. And so in order to avoid that, people pick more green fruit, which is less ripe. And so when it travels as a greener fruit, it's releasing less ethylene, and it can travel further distances. But it's not as yummy. That's why fresh fruit is always the best.
Yeah, for sure.
That was an aside, I'm sorry. But then once it starts ripening, it is releasing ethylene. And it is involved in development, germination, abscission, and stress response. So it's all over the place, and it's, I guess if I had to say, it's like a hormone that also speeds up processes.
For sure.
It's hard to put into words, and also I don't know the whole story. That's my best guess. But those were the main 5 hormones that have been known for a while, and then we've recently classified 5 more.
Okay.
And we're good on time.
We got plenty of time.
We'll jump into those. And so this is fun because ethylene is a gas.
Yeah.
So this is a form of communication that is plant to plant. So a lot of communication happens on a cell level, and some of it happens on tissue level, and then organ level. So it can be like the whole leaf responds to this, the whole stem responds to this, and maybe the whole plant responds to this. And then we move into gases, and I'm going to tell another plant to do this.
Sure.
And this is the most interesting in, to me, jasmonic acid, or jasmonate. Whatever.
Fair enough.
And salicylic acid, which you may have heard because it is something that we use. It's an aspirin.
Yeah.
It's a pain reliever. But these 2 are hormones that mediate defense.
No kidding.
So, but it's 2 different types of defense. So jasmonic acid is response to an outside attack. So if I get hit with a fungus or something is chewing on me within the plant, I'm going to send jasmonic acid out from that wound, and it's going to tell other leaves that aren't being chewed on right now to start prepping the defense.
So other parts of the same plant. No kidding.
And it becomes, and basically that defense is like the plant kind of secretes these things that inhibit insect digestion, or they break down fungi cell wall. I mean, this is another thing about plants, is that they have like these ranges of tolerances and things that they're adapted to. So they're just always trying their best to live.
Definitely.
And so like,
and producing those defense compounds, like that's an energy expense, that's a material expense that you don't necessarily want to be putting forth all the time. Wouldn't it be nice if you just knew, oh, now that I'm getting eaten, I know that I should make this in other parts to not get eaten.
And that's a great point. Because a lot of research is called, like they do research on like constitutive gene expression, which just means the gene is always turned on.
Sure.
And so if you always turn on the genes that respond to jasmonic acid, the whole plant is defended. And it's a lot harder for the plant to even get attacked in the first place, because it's hard for an insect or a fungi to build up an attack against an already built wall. But the plant shows some reduced growth.
No kidding.
Because it's spending energy on defenses. So that's just another part of that balance. However, and we'll go into salicylic acid, which is about internal defenses. So if a bacteria gets in me, if a virus gets in me, how do I respond to that? How do I build the defenses? Salicylic acid is what builds up that. And then plants have immune systems too. So they are less able to get attacked by some things, and if you get attacked by it once and it survives, it's more ready the next time.
No kidding.
So, but these 2 are antagonistic. So a plant has to mediate resources between being internally defended or externally defended based on what's happening in the environment.
Sure.
So, and then the plant can methylate these.
Oh gosh.
And all that means is it attaches a methyl group, so another carbon, and this hormone is turned into a gas. And it can be released from the leaves. And the coolest part is it can tell other trees, hey, I'm being attacked. You need to bulk up your defenses. The insects are coming towards you next. Or I think the virus is going to spread through the soil. Or it's like, hey, buddy. I want you to live.
No kidding.
Even if I don't make it, I want you to live.
And that's wild, because it's like, when I was a kid, like growing up, that kind of stuff, that's like, oh, that's hippy dippy. You're saying plants talk to each other, but it's like, as we learn more about these hormones and as we learn more about plant interactions, it's like, well, it's like they're not opening their mouths and speaking to each other, but there is some communication going on. We are letting you know, I'm experiencing trouble. You may experience trouble yourself soon. Prepare yourselves for it. Like it's an interesting thing.
And that's a great segue into our next hormone, strigolactone. Strigolactone comes from the word strigol, which is its like main part. But what's interesting is it also was found when we were dealing with striga, which is witchweed.
Witchweed, yeah.
And I don't know like what the naming is, if striga like uses a lot of strigol, or
I
it has to be
don't know.
something like that.
Sure. But I know witchweed, it's a parasitic plant that needs a host in order for the seed to germinate so that it can then puncture that root and actually grow into it.
Exactly.
It's a real problem in some parts of the world. We've more or less eradicated it in America.
But it was a huge project to do that, and it was very important. And I think it's corn is the main host.
Yeah, for sure.
And, you know, we got to have our corn.
Yeah,
So,
absolutely. That's what they make Cheetos out of, Evan.
God, put respect on the Cheetos. So how they discovered this is they wanted to just get rid of witchweed, so they were letting fields lie fallow and hoping that the witchweed would germinate, so that way all the seed bank would just be gone and the parasitic plant would have no host and die out. But the witchweed wouldn't germinate without a host.
No kidding.
So when corn germinates and grows its roots, it sends out strigolactone. Why? It sends out strigolactone to invite mycorrhizal fungi into itself.
No kidding.
Because corn is a big host for mycorrhizal fungi.
Yeah.
And so strigolactone is the signal hormone for the fungi to say, oh, we got someone here. We got someone here who we can work with.
Definitely.
And striga, the witchweed, was using that as its germination cue. Exactly. So when it came into contact with strigolactone, boom, it germinated. And instead of getting helpful fungi, it got a parasitic plant.
Oh, no.
Yeah. And the way they further tested this, because they were like, well, maybe strigolactone doesn't just do this. You know, we always have to test things. One, it increased strigolactone production when there was nitrogen and phosphate stress.
Okay.
So a plant was low on nutrients, and so it produced more strigolactone, and that makes sense given this theory, because it's like the mycorrhizal fungi are able to reach further into the soil and pull those things to the plant.
Yeah.
And two, the strigolactone was only produced under phosphate and nitrogen stress if it was a host that is known to have like a mycorrhizal companion. So that is one, and that's not, and all these hormones, we don't know everything that they do, but that's definitely one thing strigolactone does, is it calls for root symbiosis.
No kidding.
And the last two are brassinosteroids,
Okay.
which are especially found in anthers and pollen cells and seeds.
Interesting.
And it's almost not in vegetative tissue at all. It's in super, super low concentrations. Dennis actually did his paper on cytokinin, so he knows a lot about hormones. But one of the reasons this wasn't well researched is because you need buckets and buckets of plant material just to get a little bit of brassinosteroids, so it's hard to research.
Yeah.
And but anyway, once they kind of figured it out, it has to do with pollen germination and like just things happening at the right time in regards to sex cells. And then it also is the secret like commander in chief of cell division. I think it was cell division.
Okay.
Because if a plant is mutated to not produce any brassinosteroids, it doesn't matter how much auxin or cytokinin you apply, it won't increase its cell division at all.
No kidding.
Yeah. So they interact in some way that we don't really know.
For sure.
But brassinosteroid is like the tiny but big boss of cell division. Without it, auxin and cytokinin have no effect.
No kidding.
Yep.
That's wild.
Yeah. So it's all about the pollen tube, essentially, as well as one of the big things. So when a pollen grain lands on the stigma, it has to it's one cell, and it has to make this super long cell to reach the ovary. And so brassinosteroid manages that. And it's also, I think, involved with a plant is allowed to be self-incompatible. Yeah. So it knows, it recognizes if its own pollen grain landed on that, and it's like, I won't let you. Yeah. And brassinosteroids are also mediating that.
No kidding.
And the last and final hormone are just peptides. And this is really confusing because it's just a string of amino acids. Sure. And it's like, how can you call that a hormone? It's just a string of amino acids. But they do very specific things, and they travel far across the plant. One example, there's like 30 that have been found, is cysteamine, it's called. And so this is part of like jasmonic acid,
Okay.
where it'll disrupt insect herbivory at sites further away from infestation. So it's a defense prepper.
No kidding.
Oh, and cysteamine regulates self-incompatibility. Sorry, I mixed that up. But I think brassionoceras are also involved.
What's wild to me about this is like we learned those first 5 plant hormones, and I promise you, the botanists that figured that out were like, we got this. We completely understand plants. But then it's just like the more you look into it, the more there is to learn about them, really, and the more you discover. So I love how like we just stumble upon a whole new class of plant hormones, and there's just way more to understand about them. It really fits into your thesis of these things are crazy. There's so much going on here that we just don't know.
Yeah.
But it's just so fascinating how much there is to explore and discover in them.
Yeah, and it's such a common question is how does the plant know?
Yeah.
And it's like the plant doesn't know. It's just like you can't oversimplify it by saying the plant is just responding to its environment. But
Sure.
it's like through this amazing, beautiful system, it understands what's happening in its environment, and it does the thing that it needs to do.
No kidding.
It's at the end of the day, it's very simple. You can look at a plant and say, well, it's growing. And yes, you're right. And today I just wanted to shed some light on some of the weird and wacky stuff going on inside the plant.
For sure.
Because I see this and I'm a little awestruck.
Definitely. As well you should be. Like there is so much to it. And it's like you really can go really deep into the weeds, really deep in the nitty gritty on this, but just like highlighting just how complicated and how amazing it is, I think is just really valuable. Just letting you know there is more out there to explore and discover for yourself. You can devote the rest of your life and your next life to the study of all of this, and you still won't get all of it for sure.
They say the world outside expands infinitely and the world inside expands infinitely.
Oh,
so as you zoom in on the inner mechanisms of any organism, or even your own mind.
Whoa. No, for sure. Infinity of scale. Like I love that. Yeah. Cool. Well, Evan, we did have a couple of questions come in. Jared had a very good one. He said, this is super interesting. I'd never heard of abscisic acid before. If it stimulates root growth and closes off the stomata, is there such a thing as a synthetic form of this that could be used to reduce transplant stress or drought stress? Like, is there a way to apply these things to sort of circumnavigate problems to plants that we bring on them ourselves?
That's a great question, and one of the most common application of these sort of like advanced sciences is in the form of herbicides, growth regulators, and promoters. Which we don't use here because our job is to set the plants out, and also it's mostly used on crops, systems where you have to do a lot of control. There are
horticultural crops where it is actually part of the regimen, like you have to put in poinsettias, for example. Poinsettias is the one thing that I'm thinking of. Like you have to apply plant growth regulators to poinsettias if you're going to get them at the right size, to get them flowering at the right time so that people will actually buy them when they're doing their Christmas shopping and that kind of stuff. And so they do prescribed doses of plant hormones in those production settings to just make sure things are doing what they need to do at the right time. So I think Jared, in theory,
I was
go for it.
I was only half listening. I was formulating my answer.
Go for it.
I think the message I want to convey is that the plant really knows what it's doing. Yeah. And so I think it would, in 99.9% of cases, be a mistake to even apply abscisic acid. Because the plant is producing it and it has all these equilibrium states it's trying to reach, and so when it knows something is out of whack, it's already started doing the things it needs to do.
Correct.
So I would say that we are not the best eyes. The plant probably knows what it's doing better than we do.
If you're just a gardener out in your garden hoping to make the happiest, healthiest plants, then you probably should avoid doing this. However, if you're a grower who wants to force these plants to grow in this environment that's really conducive to disease or really conducive to, they're just going to grow way too much and get too big to sit on shelves. Like if you're putting them in a place where they're not going to do what you want them to do, then it's like, then you need to consult the literature, consult the research, make sure you're doing things at the prescribed way at the prescribed time, and like then you're really dialing in the science on it there. But if you're just a gardener out enjoying your garden, like let the plants be the plants. Like they know better than we do how they grow, honestly. And for sure, we learn from them.
Yeah, and I would say if a plant is not meant to survive our drought, sometimes that's okay too.
Sometimes that's okay too. Yeah. No kidding. Like there are ideal times to transplant and there are less ideal times to transplant. But like even if you're doing it at the ideal time, doesn't guarantee success.
I forgot about that that part of the question. Decreasing transplant stress. Yeah. I mean, there might be things that you could do before transplanting to kind of prep the plant, because transplant is a huge shock. You're right. It's a very sudden and human mediated activity.
For sure.
So a plant can't really prepare for it.
Well,
so actually, that might be a good point. I forgot about that part of the question. I'm sorry.
No, you're okay, because one of the ways to increase the success of transplanting is to go through a year before and like trench around the plant to like cut the ends of the roots so that it starts growing new roots back there. So it kind of, that sets the natural processes of the hormones to start balancing themselves so it can prepare for that kind of thing. Like that's a way you can cue it up. But again, like if you're trying to dial it in with the plant hormones themselves, that's going to get a little more complicated. There's inch by inch, life's a cinch. There are ways to make things happen without resulting to doses of plant hormones. I assure you. Cool.
Good question.
Absolutely. Fantastic question. And I love that you're thinking about like the practical applications of these things, because there are practical applications of these things in production settings, probably incredibly impractical and bizarre if you're doing it in your own home landscape. For sure. Okay, so Gail she needed a bit more information about the burled wood. She's saying, is burled wood, is that a result of signals of plant hormones, or is that mostly the result of the damage from the bacteria causing that?
So it's the bacteria abusing the plant's hormone production. It sends its own genetic information into the plant.
Yes.
To overproduce these hormones. So it's caused by the bacteria, but it's abusing the plant system. Does that answer the question?
I think so. It's not a happy system within the plant. We know that much. I think like the genetic information from the organism is absolutely disrupting the balance of hormones in there. So it's not necessarily, it's not like doing an attack on the hormones, but like the attack that it's doing is disrupting the system.
Yeah, and it's a localized infection, so it's not really affecting the plant that much. If anything, it's just ugly, and there's nothing you can really do about it, because even if you cut it clean, that's not really great for the plant because it's a really weird wound to close. Yeah. And it's also the bacteria is probably still there, and you've only probably increased the amount that it can sort of infect that area. So that's just how it is.
Okay. And Jared had a follow-up comment to his question. He said, I guess I was getting at something like, could it be added to a product like a spoma biotone starter, so it's like a one-stop shop for planting, like adding rooting hormone to your potting mix in order to encourage root growth? I imagine that's been studied, and I imagine it didn't work. Like it seems like an obvious thing to try to put rooting hormone in the substrate.
The other thing about hormones is they do their best work in plants, but also if we're applying them on small plants. So when we're propagating it, we're just putting rooting hormone on a single cutting. Yeah. So I think the amount of stuff you'd have to apply on a whole plant, both with abscisic acid and rooting hormone, I don't think it's economical. Right. And I think there's better practical things you could do. I forget the exact word, cultural things that you should be doing to kind of make your plant a success, rather than applying exogenous hormones.
Definitely, because one thing about taking cuttings, for example, there are lots of species of plants that you take a cutting, you skip the dipping in the rooting hormone. Like there is enough rooting hormone being produced by the apical meristem of the cutting itself that you just stick it in dirt and it roots easy peasy. Like it's not necessarily like the presence of rooting hormone. It's not like we need to add rooting hormone from outside the system. Like it's already in there. It's just making sure the balance is such that it's eliciting the response that you want it to elicit.
Yeah. And not to crush your curiosity or anything, because I'm sure like all these things could continue to be studied, and we don't know. So
yes,
there's probably an application for it.
Yeah, and Jared says he's just a curious person, but he's definitely not going to go out and buy a 55-gallon drum of ABA. So no worries, Evan, you're okay. You're inspiring, but not that inspiring. It's okay. Cool. Okay. Well, that is all of the questions that came in today and all of the time that we have for today anyway. So that's fantastic. Thank you, Evan, for taking us through this. Like it's a big topic and it's just a lot to go through, but your enthusiasm and your interest in the topic, I am certain was infectious. So we definitely appreciate you sharing your passion for plants with everybody here today.
Yeah, and let me know in the comments if you'd like to see more of this stuff, because I really enjoyed talking about this stuff. It's not that practical, but if you guys are interested, I would love to continue to share this interest. So yeah. Absolutely. Thank you for joining us.
Absolutely. Thank you, everybody, for joining us on the Midweek program today. We will be back next week. Ooh, yeah. Yeah, next week we're going to be talking about therapeutic horticulture. We're going to be joined by Miss Amy Bruzacchesi, who's very well involved with the Therapeutic Horticulture Network at NC State. So it should be a really wonderful program. We hope we will see you all next time. Y'all take care.